Biomedical research and the development of new treatments often use laboratory-grown human cell cultures. Among the many cell lines, one of the most famous is HeLa, uterine endothelial cells. These cells, which imitate a simplified human in laboratory studies, are eternal. They can divide endlessly, endure decades in the freezer, and can be divided into parts in different proportions. These cells found their way into big science completely unexpectedly. They were taken from a woman named Henrietta Lacks, who died shortly afterwards. This is biotech science, and here is the first line of immortal human cells. Henrietta Lacks lived in the small town of Turner in Southern Virginia with her husband and 5 children. On February 1st, 1951, Henrietta Lacks went to Johns Hopkins Hospital because she was concerned about strange discharge that she periodically discovered on her underwear. The medical diagnosis was terrible and merciless: cervical cancer. 8 months later, despite surgery and radiation exposure, she died. While Henrietta was at Hopkins, the attending physician sent her tumor for analysis to George Gay, head of the Tissue Cell Research Laboratory at Hopkins. Cells from the tumor were removed without her knowledge or consent. It is worth noting that at that time, culturing cells outside the body was only in its infancy, and the main problem was predetermined cell death. After a certain number of divisions, the entire cell line died. George Gay discovered that they could support life. He was able to isolate one specific cell, multiply it, and create a cell line. George named them HeLa cells, after the initial letters of Henrietta Lacks' name. These were the first human cells grown in the laboratory that were immortal. They did not die after several divisions and could be used in many experiments. Their growth suppression program turned off after a certain number of divisions, that is, programmed cell death did not occur, unlike ordinary cells that have a Hayflick limit. This happens because, as with many types of cancer, HeLa cells produce the enzyme telomerase, which extends telomeres at the ends of chromosomes. Since 1951, HeLa cells, being in an artificial environment, have been dividing to this day. Moreover, the total mass of the offspring of these cells is several tons, and their age is already 100 years older than Henrietta herself. The peculiarity of these cells is the fact that they divide twice as fast as normal cells and at the same time are unpretentious and do not require special growth substrates. These cells adapt very quickly to conditions and tolerate cryopreservation well and remain frozen for decades. The processes of cell cryopreservation were also studied for the first time on this cell line. Thanks to HeLa cells, the first inactivated polio vaccine was developed by Jonas Salk in 1953. Especially for this purpose, large-scale production facilities for the production of these cells were created for the first time. Their use made it possible to accumulate in a short period of time a huge number of virus-sensitive cells necessary to obtain the required amount of vaccine. Subsequently, these cells were used to isolate and cultivate a number of other viruses, for example, HIV, coronaviruses, Ebola, measles, and herpes. Thus, HeLa cells contributed to the development of virology. For a long time, scientists believed that human cells contain 48 chromosomes. In 1956, scientists found that humans have not 48, but 46 chromosomes. These cells were among the first biological objects to be repeatedly sent into outer space since 1960. After returning, it was discovered that they were able to tolerate weightlessness well. In 1972, research was carried out that led to the creation of the first recombinant DNA molecule. These cells were used in the development of in vitro fertilization technology and also found use in the process of cloning higher organisms, including Dolly the sheep. The HeLa cells were infected with the papillomavirus from the start, which often happens to cancer cells. Thanks to HeLa, a connection between the human papillomavirus and cervical cancer was identified, and the role of telomerase in preventing aging and cancer development was studied. In addition, they were used directly as biological objects in the compilation of a genetic map in the Human Genome Project and became the origins of the development of cellular engineering. Currently, the cells are used by millions of laboratories around the world and make it possible to study the mechanisms of carcinogenesis and search for anti-cancer drugs. These facts directly indicate that cancer cells, which have been cultivated for 70 years, were ultimately able to save and continue to save a huge number of human lives.